Welcome to Deep Dive Lab, where we break down and dissect cutting-edge scientific research, from advanced biomedical technologies to breakthroughs in AI, material science, chemistry, and physics, all told as compelling, easy-to-follow stories. And today, we are looking at something that, honestly, it kind of fundamentally changes how we think about human biology.
Oh, absolutely. It's a total paradigm shift.
Right. So, I mean, imagine, if you will, that your DNA is a massive multi-volume instruction manual for building you. It has all the blueprints, all the text. But, here's the thing. Having a manual doesn't mean your body actually reads every single page.
Yeah, not even close.
Exactly. So who or what decides which pages get, brightly highlighted for daily use and which pages get permanently glued shut so they can never be read? Well, that is the fascinating world of epigenetics. And today, we are taking that concept and applying it to a really intriguing mystery. So here's a strange question. Why does alcohol affect people so differently? And how does heavy drinking actually leave a physical molecular fingerprint on our biology? Okay, let's unpack this.
Yeah, it is a mystery that has frustrated the scientific community for decades, really. And to understand why solving it is such a big deal, we kind of have to look at the sheer scale of the problem.
Right.
Because alcohol use disorder, which most people know as AUD, is just incredibly common. It affects roughly 10% of the population at some point in their lives.
Wow, 10%. That's huge.
It is. And through decades of meticulous twin studies, where researchers compare identical twins who share all their DNA to fraternal twins who only share half, scientists have long known that AUD is about 50% heritable.
Okay, wait. So meaning half of your risk for developing this disorder comes down to basically the genetic hand you were dealt at birth.
Exactly. Yeah.
Right. So that is the literal text written in that massive DNA instruction manual we just talked about.
Yeah.
If you have the genetic vulnerability, the instructions for AUD are just sitting there in your manual.
That's it. But the pure DNA sequence itself, like the actual letters, the A, C, T, and GS, it doesn't tell the whole story, because you can have that severe genetic vulnerability. You can have the instructions right there in your manual, but, never actually develop the disorder.
Because the page stays glued shut.
Precisely. Scientists knew that environmental factors, things like your lifestyle, your daily stress levels, early childhood trauma, and just your basic exposure to alcohol, they play a massive role in whether those genes actually do anything. And they exert this influence through epigenetics, specifically a molecular mechanism called DNA methylation.
Okay, wait, let's break that down for a second, because I feel like we hear the word epigenetics thrown around a lot. What is DNA methylation actually doing on a physical level? When we talk about highlighting pages or gluing them shut, what is the actual biological glue?
So it's quite literally a physical roadblock. A methyl group is just a tiny chemical tag. It's literally one carbon atom attached to 3 hydrogen atoms. And your body takes these little chemical tags and physically attaches them to very specific spots on your DNA strand.
Oh, wow. So it's actually sticking to the DNA.
Yeah, exactly. It doesn't change the underlying genetic sequence, right? The letters are exactly the same. But by sticking this bulky little chemical tag onto the DNA, it physically blocks the cellular machinery from reading that section. The machinery comes along, hits the methyl tag, and just stops.
Oh, I get it. So that specific gene gets turned down or silenced entirely.
Yeah.
It's like putting a physical piece of tape over a line of text. I mean, the text is still there, but you can't read it, so you obviously can't build whatever that line of text was instructing you to build.
That is exactly right. So knowing this, researchers naturally started looking for these methylation tags, this chemical tape in people with alcohol use disorder. They conducted what are called epigenome-wide association studies.
That is a mouthful.
Yeah, in the field, we just call them EWAS for short. And they would take a group of people with AUD, scan their entire genome for these methyl tags, and basically just try to find a pattern. But the early results from these studies were, frankly, deeply confusing.
Really? How so?
Well, one study would point to a specific set of tags, and then another study, maybe a few months later, would come out and point to a completely different set of tags. They were just completely inconsistent.
Okay, I think I see the problem. Because if genetics are the piano keys...
Then epigenetics is the sheet music telling you how hard to hit them.
Right. So earlier scientists were essentially trying to find the definitive sheet music for AUD, but it sounds like they were all listening to different bands in completely different venues, playing entirely different songs. Why was it such a mess?
The primary culprit was sample size. These early studies were only looking at very small groups of people. I mean, we're talking ranging from maybe 198 individuals to a maximum of about 11,132 people.
Oh, wow. Yeah, that's not a lot.
No, it's really not. Especially when you are dealing with a condition as complex as alcohol use disorder. You have people with vastly different severities of the disease, completely different dietary habits, life circumstances, genetic backgrounds. A small sample size is going to generate a massive amount of statistical noise. There are just too many variables muddying the waters.
So if you only look at, like, 200 people, you might accidentally think a specific epigenetic tag is caused by alcohol, but it was actually just because half the people in your small group happened to be smokers or shared a similar diet or something like that.
Precisely. The real signal just gets lost in the noise. Which brings us to the crux of the problem. This raises an important question. How do we cut through all that statistical noise to find the actual truth?
Right.
How do we find the core biological signature of AUD that is consistent across different ages, different genetic backgrounds, and, different severities of the disorder?
Well, okay, I mean, if small sample sizes are creating the noise, I'm guessing the only way to solve the problem is build a massive sample size.
You would be correct. And that is exactly where a landmark study recently published in the journal Nature Mental Health comes in. It was spearheaded by Dr. Leah Zilich and her colleagues at the Psychiatric Genomics Consortium.
Okay, so what did they do differently?
Well, they knew they couldn't just run another small study. So they conducted a massive meta-analysis. Instead of starting from scratch, they combined the data from 7 different already established cohorts. They pulled in studies from the United States, from Germany, and from the Netherlands.
Oh, nice. Okay, so they cast a much wider net to capture way more diversity.
A huge net. And what's really interesting is they included groups that were specifically recruited for AUD treatment, sure, but also groups recruited for trauma or depression, which, as we know, frequently occur right alongside AUD.
Yeah, that makes sense.
So by pooling all this international data, they gathered a sample size of 3,775 individuals, and critically, 1,325 of those individuals had a formal clinical diagnosis of alcohol use disorder.
Wow. Okay, that is a massive leap from 198 people. With nearly 4,000 people's genomes mapped out, the statistical noise should theoretically cancel itself out, right, leaving just the actual signal. So what did the universal sheet music actually look like?
For the first time, they found real clarity. I mean, out of the millions of possible places on the human genome where a methyl tag could attach, they discovered 118 specific CPG sites that were significantly and robustly associated with AUD across all these diverse populations.
Okay, I want to clarify something quickly for everyone. A CPG site, that's just the specific biological parking space where these methyl tags are allowed to attach, right? It's like where a cytosine and a guanine base sit right next to each other on the DNA ladder.
Exactly. You can't just put a methyl tag anywhere willy-nilly. It has to be at one of these CPG sites. And out of all the millions of parking spaces, these 118 sites consistently had different methylation patterns in the people with alcohol use disorder.
Man, 118 specific molecular tags. We finally found the fingerprint. So let's talk about what these tags are actually doing.
Yeah.
Did the researchers look at where these 118 tags were located in the instruction manual?
They did, and it is highly revealing. The absolute strongest association, like the loudest, clearest note in that sheet music, was found at a site on a gene known as LOC10/0505942.
Okay, definitely not the catchiest name in the world.
No, yeah, geneticists aren't great at branding, I'll admit that. But the function is what really matters. This particular gene produces what's called a non-coding RNA.
Okay, stop right there. Because most of us learned in high school biology that genes produce proteins.
Yeah.
They make the building blocks of the body. What on earth is a non-coding RNA?
That is a great distinction to make. You're right, most of the famous genes are blueprints for proteins, like the protein that makes your muscle tissue or the enzyme that digests your food. But a non-coding RNA doesn't make a protein.
Then what does it do?
Instead of a blueprint for a machine, think of it as a memo issued to the factory floor. It acts as a regulator, basically floating around the cell and controlling how other genes behave, turning them up or down.
Oh, that's wild.
Yeah. So a heavy epigenetic change here means the regulatory memos are just getting messed up. What's also super compelling is that this specific site has been linked in previous studies to smoking, liver cirrhosis, and type 2 diabetes.
Which honestly makes perfect sense because those conditions frequently overlap with heavy, long-term alcohol use. The biological damage is all intertwined. But you mentioned earlier that these 118 tags weren't just random, they were clustering around specific bodily function.
Yes, they were. The
Yeah.
researchers mapped these 118 tags and found they were heavily clustered around genes responsible for crucial internal logistics, things like amino acid transport and vesicle transport, which is basically how cells package materials and move them around internally.
Okay.
But one of the most notable clusters was around a gene called SLC7A5, and this gene is heavily involved in the blood-brain barrier.
The blood-brain barrier. Okay, that is the literal security checkpoint for the brain. It's a tightly packed layer of cells that stops dangerous toxins in the bloodstream from sneaking into the delicate brain tissue.
Exactly. It is highly selective. But, the brain still needs nutrients to survive. It needs amino acids to build neurotransmitters like dopamine and serotonin. So the SLC7A5 gene provides the instructions to build a specific transport protein.
Like a carrier.
Yeah, think of it as a specialized ferry boat that is authorized to carry essential amino acids across that security checkpoint and into the brain.
Wow. So if alcohol exposure is causing the body to glue these epigenetic tags onto the SLC7A5 gene, it's essentially altering the blueprints for those ferry boats. It is fundamentally disrupting the brain supply chain.
Yes.
I mean, that would absolutely change how the brain functions, how it builds neurotransmitters, and potentially how addiction takes root in the first place.
That is the working theory right now, and it just shows how physical this disorder really is.
Okay, hold on, though. I need to push back on this because there's a glaring issue here that I'm seeing.
Sure.
You just talked about the blood-brain barrier and neurotransmitters in the brain. But these researchers were looking at blood samples, right? They were extracting DNA from peripheral blood drawn from the participants' arms. But alcohol use disorder is fundamentally a disorder of the brain. It's about neurocircuitry, addiction, craving. My liver cells don't have the same epigenetic tags as my neurons. The tape is in different places. So isn't looking at blood completely missing the target?
I'm really glad you brought that up because tissue specificity is the great hurdle of epigenetics. You are absolutely right. The epigenetic manual in a blood cell looks very different from the manual in a brain cell.
Right, so how do they justify that?
Well, the researchers knew this was a massive potential vulnerability, so they utilized a specialized tool called Beacon to test it. Beacon is essentially a massive database that allows scientists to see if a methylation pattern found in living human blood correlates with patterns found in post-mortem human brain tissue.
Okay, so they check the blood tags against actual brain tissue from, like, tissue banks. Do they match?
Well, they found some solid correlations in certain specific brain areas, but if we're being completely objective, overall there was surprisingly little overlap with the results of previous postmortem brain studies on AUD.
Okay, wait, so doesn't that invalidate the blood test? I mean, if it doesn't match the brain, what are we even looking at?
Not necessarily, and here is why. We have to look at how those postmortem brain studies were conducted. Brain tissue is incredibly difficult to acquire for research, as you can imagine. Because of that, those previous postmortem brain studies had incredibly small sample sizes.
Oh, no. Don't tell me.
Yep. We are talking about studies looking at only 46 to maybe 119 brains.
So we are right back to the exact same small sample size problem, just in a different tissue. The brain data is too noisy to be a reliable baseline.
Exactly. The lack of overlap doesn't mean the blood data is wrong. It just means the brain data we currently have isn't robust enough to compare it to. In fact, many experts argue this makes the blood data even more vital.
How so?
Well, we have to remember how alcohol operates. It doesn't just bypass everything and go straight to the brain. It is processed in the gut, it's metabolized in the liver, and it bathes the entire cardiovascular system. It is a deeply systemic chemical. So peripheral blood gives us a robust, highly accessible, systemic marker of what this chemical is doing to the body as a whole over a long period of time.
Here's where it gets really interesting. Because Dr. Zilish and her team didn't just look at this one massive group of severe AUD patients, did they? They wanted to see if these signatures existed outside of clinical addiction, so they took their findings and compared them to a completely different study.
Yes, they did. They took their list of 118 definitive sites and cross-referenced it with a recent massive study called Generation Scotland. Now, the Generation Scotland study wasn't looking at severe clinical alcohol use disorder. They were just looking at general alcohol consumption in a vast everyday population. Just normal, everyday drinking habits.
And the overlap between those two lists is staggering. Out of the 118 epigenetic sites strictly linked to severe clinical AUD, an astonishing 65 of those sites overlapped perfectly with general heavy drinking in the Generation Scotland population. That is a 55% overlap.
What's fascinating here is how this finding blurs the line between biological vulnerability and lifestyle exposure. It brings us to the classic chicken and egg problem of epigenetics.
Oh, right.
Yeah, we have these 65 epigenetic marks. Are they the underlying biological cause that makes someone genetically vulnerable to AUD in the first place? Or are they the molecular scars left behind on the DNA as a result of prolonged heavy drinking?
Right. Like, did the manual come pre-highlighted, making you inherently crave alcohol more than the average person? Or did years of heavy drinking cause your body's cellular machinery to just frantically slap those methyl tags onto your DNA in a desperate response to the constant toxicity?
Exactly. And the evidence heavily leans toward the latter for these overlapping sites. Obviously, alcohol consumption is a necessary prerequisite for developing alcohol use disorder. But this massive 55% overlap strongly suggests that many of these epigenetic signatures aren't the cause. They are reflecting the systemic biological consequences of heavy exposure over time. Your lifestyle is actively writing on your DNA.
Wow. And the researchers didn't just leave that as a theory. They put this idea to the ultimate test. They wanted to see if they could actually use these molecular scars to predict behavior.
They did. And honestly, this is the part of the study that really bridges the gap between theoretical science and practical real-world medicine. They took all the data from their meta-analysis, the exact locations, the weights, and the values of these methylation tags, and they built a mathematical model. They created what is called a methylation risk score, or MRS.
It's essentially a biological breathalyzer. But instead of checking your breath to see what you drank an hour ago, it checks your DNA to see what your historical drinking habits have been over the years.
That is a very evocative way to put it, and mechanically it's pretty accurate. To test if this score actually worked, they took it to a totally independent group called the Elluariso cohort.
Okay, who were they?
This was a group of 2,534 hospital patients who had been admitted for coronary angiography. These people were not recruited for an alcohol study, they were just regular heart patients. But the researchers had their DNA methylation data, and they also had their self-reported data on how much alcohol they drank.
So if I had this right, they feed the DNA blood data of these random heart patients into their new algorithm without telling the algorithm anything about the patient's drinking habits. And the algorithm has to guess who the heavy drinkers are based purely on the epigenetic tags.
Exactly. And the score successfully predicted heavy drinking in this totally independent real-world population.
So what does this all mean? If someone is listening to this deep dive right now on their morning commute or, while making dinner, why should they care about a methylation risk score?
Well, it comes down to the numbers and what they represent for the future of medicine. When they ran the model on the Elluaric cohort, this methylation risk score explained about 2.06% of the variance in general, daily alcohol consumption, and more importantly, it explained 10.48% of the variance in heavy drinking behavior.
Okay, I have to be honest here. 10%. If my weather app was only 10% accurate, I'd delete it from my phone. If my GPS only got me to the right place 10% of the time, I'd throw it out the window. Is capturing 10% of the variance really worth writing a landmark paper in Nature about?
It is a very fair question, and frankly, it's one a lot of people ask when they look at genetic studies. But you have to remember the context. Human behavior is wildly complex. It is highly heterogeneous. The scientific term is that it's a very noisy phenotype, which just means the observable trait or behavior we are looking at. Think about why someone drinks heavily. It is influenced by their underlying genetics, sure, but also their daily stress levels, their income bracket, their cultural background, peer pressure, childhood trauma, their metabolism. I mean, there are thousands of intersecting variables.
Okay, I see where you're going with this.
Right. So to be able to capture over 10% of that incredibly complex behavioral variance from a single static blood test reading your epigenetic tags. In the world of psychiatric genomics, that is actually a crucial breakthrough. It proves that despite all those other sociological and psychological factors, our peripheral blood carries a distinct, reliable, and readable record of systemic alcohol exposure.
That frames it perfectly. We are finding a clear biological signal inside a hurricane of psychological and environmental noise. So yeah, 10% of a hurricane is a massive deal. But just to manage expectations, I assume I can't go to my doctor tomorrow, get some blood drawn, and ask for the AUD methylation test.
Not tomorrow, no. We are not at the point of clinical diagnosis just yet. The predictive accuracy of the model, what statisticians measure using an AUC-ROC curve, was 0.657.
Okay, what does that number mean?
Well, Without getting bogged down in the math, you just need to know that a score of 0.5 is basically a coin flip. It's completely random. A score of 1.0 is a perfect prediction every single time. So a score of 0.657 is solid, it is statistically significant, and it's fantastic for advancing research. But it is not quite high enough to be used as a definitive diagnostic tool in your doctor's office today.
But it's the foundation. It proves the concept works.
Exactly. It lays the absolute groundwork. Imagine a future, maybe a decade from now, where a simple blood test could help doctors diagnose AUD more objectively, rather than relying solely on self-reported questionnaires, which, we know can be inaccurate.
People definitely lie to their doctors.
They absolutely do. Or imagine using this risk score to predict a patient's prognosis in a rehabilitation setting. If we can literally see the molecular scars on their DNA, maybe we can track those scars healing as a patient maintains sobriety over time. Eventually, we could even tailor specific pharmaceutical treatments based on a patient's unique epigenetic profile.
If we connect this to the bigger picture, this research fundamentally proves that our lifestyle choices don't just happen to us. They aren't just abstract memories in our brain or a passing chemical state that gets washed away the next day. Our habits, particularly heavy alcohol consumption, actively rewrite how our bodies operate on a cellular level. They change the physical structure of how our genes are expressed.
Yes, it bridges the gap between our behavior and our biology in a way that is visible, measurable, and profound.
Let's recap this journey we've just been on because it is truly mind-bending when you look at it from end to end. First, by combining data from nearly 4,000 people across the globe, scientists finally cut through decades of statistical noise to find 118 reliable, rock-solid epigenetic signatures of alcohol use disorder.
They found the universal sheet music.
Exactly. Second, they discovered that over half of these signatures overlap perfectly with general heavy drinking, proving that there is a profound physical link between our daily habits and our foundational biology. The molecular scars are real, and they disrupt crucial systems like the blood-brain barrier.
Yeah.
And third, we can now use those very markers to create a score that actually predicts heavy drinking behavior in completely independent populations, opening the door for future diagnostic blood tests that could revolutionize how we treat addiction. I mean, it is science fiction becoming science fact.
It really is.
Which leaves us with one final lingering thought for you to mull over today as you go about your routine. If scientists can now literally read the molecular footprint that heavy drinking leaves behind on our DNA, what other daily habits, everyday stresses, or even moments of joy are actively rewriting your epigenetic instruction manual right this very second? Thank you so much for joining us on this investigation in the Deep Dive Lab. If you enjoyed unpacking this mystery with us, please follow and subscribe for more fascinating science stories. We will catch you next time.